This program is brought to you by Stanford University. Please visit us at stanford.edu.
So without further ado, I'd like to introduce Dr. Weintraub. Dr. Weintraub has a very diverse background in vascular biology. As we're going through his CV, I was quite impressed. Not only do we know him well from his work in basic mechanisms in terms of inflammation and oxidative stress and aneurysm development, but also a lot of seminal work in terms of vascular biology and in arachidonic cascade metabolites and other insights into atherosclerosis. So without further ado, I'd like to introduce Neal Weintraub to tell us about his work in oxidative stress.
Thanks very much, Phil, and thanks very much to the organizers for inviting me. And I'll have to say, I'm like the poster boy for collaborations with people helping others who are interested in aneurysm work. In particular, Alan Dougherty and Rob Thompson have been instrumental in helping my lab get going. And I want to put in a good plug for his website, as well as for Rob's lab in St. Louis, who have been just extraordinarily helpful to us. So I'm going to talk today about the role of oxidative stress in the pathogenesis of aneurysms. And everyone in here is aware of the pathology of aneurysms, characterized, of course, by intense inflammation, as Rob mentioned, associated with the structural deterioration. And the key changes that we see tend to focus on degradation of the extracellular matrix and loss of smooth muscle cells through apoptotic pathways. And this is where we became interested in the role of oxidative stress in these processes. About 10 or so years ago, it became clear that during many different forms of inflammation, that the oxidative stress occurring locally is key in activating both MMPs and inducing smooth muscle cell apoptosis. And that's what led our group to begin exploring this hypothesis, which we've laid out in a review paper we published a year ago in ATVB. So our belief is that the inflammatory milieu of aneurysms, of course, is key, very much dependent upon inflammatory cells such as macrophages and other types of inflammatory cells. These inflammatory cells, when they're recruited to the lesions, generate lots of reactive oxygen species and reactive nitrogen species. At the same time, they release proteases and they generate lots of cytokines. And all of these things together contribute to smooth muscle cell death. Moreover, the smooth muscle cells, when they become under stress, both from these pathways as well as the hemodynamic effects of tissue remodeling, generate oxidant species as well. In turn, the oxidant species can upregulate adhesion molecules to amplify the recruitment of inflammatory cells. And so the consequent activation of MMPs resulting in reduction of elastin from degradation, as well as the death of smooth muscle cells, causing an impairment in production of elastin. And in addition, you can see that these species may trigger hypertension, and so this may also link the biomechanical properties to the oxidative processes, which in turn may contribute to tissue remodeling and expansion. So a number of years ago, this hypothesis was thrown around, and there were some early papers published primarily by Mike Dubik's group way back in the 1980s and 90s, and these were largely descriptive in nature, and they examined aneurysm tissues and saw increases in lipid peroxidation products. They saw reductions in antioxidant enzyme levels, and all of these suggested a possibility that oxidative stress was locally involved. Likewise, there was some early evidence that reactive nitrogen species could be involved in this process, and then there was an interesting report published some years ago suggesting reductions in vitamin E levels specifically in patients with aneurysms, but all of these were correlative and not causal. And then studies emerged dating back about 10 or so years now, looking at this hypothesis in animal models, and a number of studies from many labs now have confirmed that there's evidence of induction of oxidative stress during formation of aneurysms experimentally in several different models. Ron Dahlman's lab showed, using his AV fistula technique, that flow loading induced antioxidant gene expression and attenuated aneurysm formation. And to my knowledge, his lab was the first to show that actually treating with vitamin E could ameliorate the aneurysm process in his model. We also showed that treatment with the antioxidant vitamin E could ameliorate aneurysm formation in the angiotensin II model. Of course, there's not a lot of good news about vitamin E, either clinically or experimentally. And in fact, in the data that have been published to date, including our data, the effects of vitamin E are rather marginal, or I would say modest at best. It requires very large doses. And a recent paper published last year suggested that the impact of vitamin E may even be less effective in older mice, which further suggests this would not be a promising therapy for humans. So free radical biologists tend to take this kind of an approach to dissecting out mechanisms in which oxidative stress may be involved in a biological process. And I apologize about the complexity here, but there's really no other way to sort of detail the different reactions that may be present, and I'll focus on some of these in detail. In particular, I'm going to talk a fair amount about this enzyme, NADPH oxidase, which is probably the key enzyme involved in generating superoxide in the blood vessel wall. This is also the enzyme present in phagocytes that's responsible for generating superoxide, and there's a lot of evidence to support the involvement of this enzyme in aneurysm formation. As you can see, particularly relevant to the phagocytic pathway, generation of hydrogen peroxide from superoxide results in the substrate from myeloperoxidase, this enzyme present in phagocytes, which in turn generates hypochlorous acid. And this is a powerful oxidant species that I'll talk about at the end of my discussion today. In addition, there's new evidence suggesting that this enzyme, PGE2 synthase, which is in the eicosanoid family of enzymes, is actually involved in oxidative stress as part of the mechanism of aneurysm formation. Gib Upchurch, who is not here today, has some evidence suggesting that manganese SOD, which catalyzes the dismutation of superoxide in the mitochondria to hydrogen peroxide, which subsequently leads into the cytosol and can affect many different cellular signaling mechanisms and has its own pathways of decompensation, could be involved in this process. And then finally, there's controversial data on nitric oxide synthase. Of course, the inducible form of nitric oxide synthase generates lots of nitric oxide, which when it's in an imbalanced state can be pro-oxidative. It also reacts with superoxide to form peroxynitride, a very powerful reactive nitrosylating species. And so there's at least some data to suggest this could be involved. I apologize about the confusion. I promise you I will try to focus it a little bit more carefully on these individual pathways. Now, why do we think this NADPH oxidase is so important? Many people in vascular biology see this as sort of in the center of a whole bunch of biological factors involved in vascular disease. And the evidence linking NADPH oxidase to vascular disease goes far beyond what I'll talk to you today about aneurysms. There's lots of evidence in atherosclerosis and hypertension, as well as several other processes. But a lot of the factors that come into play in the milieu of the aneurysmal tissue all have the same thing in common. They can upregulate and activate this enzyme. Moreover, my lab showed that reactive oxygen species themselves, including hydrogen peroxide and various lipid peroxidation products that are generated from NADPH oxidase, can actively feed forward and trigger the activation of the enzyme, so sort of setting up an amplification mechanism that once you get activation of this enzyme and generation of oxidant species, that it amplifies itself and it propagates the oxidative reactions. This is the structure of the NADPH oxidase, and again, I apologize about the complexity, but you should know that there are both membrane-bound subunits and cytosolic subunits. The membrane-bound subunits are these two, the NOx isoforms, and I've listed 1, 2, and 4, and it depends on which cell type that the prevalent isoform would be present. In phagocytic cells, it's primarily NOx2, whereas in vascular cells, it tends to be NOx1 and NOx4. P22-FOX is the other membrane-bound subunit. And these cytosolic subunits, when the enzyme gets activated, they translocate to the membrane to form the active complex. And I'll specifically talk about this one, P47-FOX. Now, I've shown several things that happen. In the phagocytes, the NOx2 isoform tends to convert molecular oxygen to superoxide, which is released extracellularly. Whereas in the vascular wall cells, the NOx1 and 2 seem to direct it more towards an intracellular release where it may be more involved in signaling. So now let me start with where we began in our own work looking in human aneurysm tissue. And I started this work when I was at the University of Iowa, and I have since relocated to Cincinnati a couple of years ago, but much of what I'll show you did come from the work that we did at Iowa. We started out by looking at human aneurysms, and what we did that was a little unique is we compared human aneurysms with the adjacent non-dilated aorta. And to the best I can tell, this was the first time this had been done. Most of the studies that I was able to identify compared aneurysms to normal aorta or aorta from another individual. And of course, there's so many confounding influences. So we were able to take the aneurysm tissue that was resected at the time of repair, the adjacent non-dilated aorta, and look in situ at superoxide and oxidative stress. What you're looking at here is a dihydroethidium confocal microscopy, and this is a technique looking at superoxide in situ using this specific probe. And we were shocked to see that in the aneurysm segments, there was a tremendous amount of superoxide seen throughout the tissue, much more than what we saw in the adjacent non-dilated aorta. And we quantified this using a different technique and showed that it was several-fold higher in the aortic section locally as compared to the non-dilated adjacent aorta. Looking at the tissue pathology, you can see under low power this area of medial degradation where there's a lot of inflammatory cells here, degradation of elastin fibers, and in that same region we saw large amounts of superoxide. I guess a smoking gun, if you will, that the oxidative stress is at least locally associated with the disease pathology. In investigating the enzymatic source of the oxidative stress, we focused on NADPH oxidase, and as you can see from these data using a NADH and NADPH stimulated chemiluminescence assay, we saw about a 2.5-fold increase in NADPH oxidase activity in the aneurysmal section as compared to the adjacent non-dilated aorta, and you can see this was blunted by DPI, which is a chemical inhibitor of this enzyme. We looked for expression of the various isoforms of this NADPH oxidase, and what came out was a large increase in expression of the cytosolic subunit P47-FOX in the aneurysmal tissues as compared to the adjacent non-dilated aorta. So in order to address the potential role of this enzyme in the pathogenesis of aneurysms, we took P47-FOX knockout mice, and we bred them into the ApoE mouse, and using Alan Dougherty's experimental model of angiotensin II-induced aneurysm formation, which I'm sure you all now are very familiar with, we implanted these mini pumps, infused angiotensin II, and then we looked at the formation of aneurysms in both ApoE mice that expressed the wild-type P47-FOX and the double knockouts, ApoE P47-FOX knockout mice. And as Alan has alluded and others in the earlier presentations that you all heard, we and others tend to see divergence in the effects of maneuvers to look at aneurysm formation and atherosclerosis. And for example, deletion of P47-FOX had no impact on the lipoprotein levels in these mice, and really virtually no impact on atherosclerosis at the level of the aortic root. Again, suggesting these disease processes may be quite distinct because, as you can see in this slide, deletion of P47-FOX had a very dramatic effect on suppressing aneurysm formation in this ApoE infusion model, and this is the worst one we saw in the entire series of experiments that we did in the P47-FOX knockout mice. This is the incidence data of aneurysm formation in the wild-type mice, the ApoE mice. You can see it's about 90%. In the mice that were deficient in P47-FOX, the incidence was only 16%, and we also saw reductions in thoracic aortic aneurysms as well. These are the size data that we found. As you can see, infusion of angiotensin II results in a large increase in aortic diameter and aortic weight, and these are significantly blunted, not eliminated, but significantly blunted by deletion of P47-FOX. Looking at the tissue pathology, I think you can see on this H&E stain the pronounced inflammation and remodeling of the aortic wall seen in the ApoE mice. Notice in the P47-FOX deficient mice, very strong preservation of the normal architecture of the aortic wall here. Likewise, we performed immunostaining for macrophages and nitrotyrosine, which is a marker for oxidative stress, and these were severely attenuated in the mice in which P47-FOX was deleted. And finally, we showed a reduction in MMP2 activity in the mice that were deficient in P47-FOX. So together, these data strongly pointed towards a role for this particular enzyme, at least in the Ang2 infusion model. Now, very recently, I note that Tim Baxter's group has published a paper that's actually still in press, I believe, in atherosclerosis. But they looked at the impact of aposinin, which is an oral inhibitor of the NADPH oxidase, and actually targets the same subunit, the P47-FOX subunit that I was talking about. And this is in the calcium chloride induction model that you've heard a little bit about. And note that in the control animals that received no aposinin, there's an increase in diameter of the aorta following application of calcium chloride. And this was abolished by treating the mice with aposinin in the drinking water. Also, you can see the degradation of the elastin fibers here in the control mice. They were preserved in the aposinin-treated mice, and this was also seen in conjunction with an inhibition of the activation of the matrix metalloproteinases that occurred in the control mice. So together, I think these data strongly point towards a role for the NADPH oxidase in the formation of aneurysms. Now also, Karl-Heinz Krause's group has recently published a paper looking at the impact of deletion of this isoform of the NADPH oxidase, NOX1, in aortic dissections. And so they studied the Ang2 infusion model, but they did not study hyperlipidemic mice. They were studying mice in the C57 background, and so they were really looking more at what they called dissections than aneurysms. And recall, this enzyme is expressed primarily in the vascular wall cells. So this may be relevant to Alan's data with the smooth muscle cell specific deletion of the AT1A receptor. And so in their study, they infused angiotensin II in wild-type mice. The Knox knockout mice is shown here. In the wild-type mice, they had 26 that entered the study. 2 died of dissections that were fatal, and they saw 6 out of the other remaining mice that developed dissection microscopically. In the NOX1 deficient mice, they had no deaths, and only 1 animal had dissection out of the 25 that they studied. So this was a marked reduction in the incidence of aortic dissection resulting from Ang2 infusion. And since the NOX1 is expressed primarily in the vascular wall cells, this may be part of the mechanism linking angiotensin, the AT1A receptor, to the downstream pathology, perhaps through NOX1-mediated reactive oxygen species. Now, these investigators also conducted microarray studies to try to understand the mechanism whereby deletion of NOX1 was protective, and they really didn't come to a firm conclusion. But what they did find, interestingly, was that in the NOX1 knockout mice, when they infused with Ang2, they saw a large increase in thrombospondin 4 expression. They don't really know what it's doing, but the increase was so dramatic, they suspected that this may be a protective mechanism that emerged upon deletion of NOX1. Likewise, Ang2 infusion in the wild-type mice increased TEMP1 expression, and this was further increased in the NOX1 knockout mice, both at the RNA and the protein level. So it could be that when you delete NOX1 and you remove those ROS, you get a counter-regulatory mechanism that emerges that then protects the animal from the dissection. Now I want to talk a little bit about nitric oxide synthase. Of course, the inducible form, the iNOS, is important in a lot of inflammatory processes, and as I said earlier, this generates large quantities of nitric oxide. It can also become uncoupled from its substrates and generate superoxide, which together can form peroxynitrite, and it's a very powerful nitrosylating species. There's evidence that iNOS expression in nitrotyrosine, which is a marker of amino acid oxidation induced by peroxynitrite, have been detected in human aneurysms, but not in normal aorta. And then in one interesting study by Tilson's group, which was really just an abstract, but they reported very high nitrite levels in human aneurysms at levels that potentially could lead to matrix degradation. So all this together was intriguing and suggested a potential role for iNOS in aneurysm formation. In an early study published by Johanning back in 2001, they use a chemical inhibitor, Amino Guanidine. It's a non-selective NOS inhibitor and has other properties as well. And they were able to show that when they gave their mice Amino Guanidine either postoperatively or even more impressively preoperatively, they could reduce the formation of aneurysms in elastase infused rats. And that was the first report. The same year, Rob Thompson's group took advantage of the iNOS knockout mice, and in their model of elastase infusion, they looked at the impact of deletion of this particular NOS isoform in aneurysm formation. And interestingly, they saw in the male mice really no impact of deletion of iNOS on aneurysm formation. Very definitive. You can see the incidence rate here, 40% in both groups. The size of the aorta virtually identical, whereas in the females, actually, deletion of iNOS was detrimental. It seemed to cause a larger dilation of the aorta in the female mice. And now, once again, Tim Baxter's group has just published a paper showing that in the calcium chloride model, they saw nice induction of iNOS in their C57 mice, as you can see here. Interestingly, when they looked at the iNOS knockout in this model, what they saw was the degradation of elastin fibers induced by the calcium chloride application was largely preserved in the iNOS knockout mice here. Likewise, the induction of MMPs in response to calcium chloride application was inhibited in the presence of the iNOS, in the absence of the iNOS gene. And then finally, the aortic diameters resulting from calcium chloride application were blunted or attenuated in the iNOS knockout mice. So I think we're in a situation where this likely reflects the difference in the models, and this is part of the morning discussion that was held in which Rob and Alan both talked about models. And it's interesting to see the different results that one can get from these maneuvers in different models. So I think the jury's still out with iNOS. Now I'll talk a moment about PGE2 synthase. There's been a lot of interest in the cyclooxygenase pathways in aneurysm formation. PGE2 has many properties that can influence smooth muscle viability and other steps that could be important to aneurysm formation. Garrett Fitzgerald's lab has recently published a paper looking at the impact of deletion of the microsomal PGE2 synthase 1 gene on aneurysm formation in the Ang2 infusion model. Now, for those of you who don't know much about icosanoid metabolism, PGE2 is the major icosanoid produced in the vasculature. And so it's interesting that when you you look at the impact of deletion of this particular gene, this may very much shift the balance of icosanoid production in the aortic wall. And it may be hard to exactly understand how this could relate to oxidative stress, but I'll try to explain it in a moment. First of all, note the impact of deletion of this PGE synthase gene in the LDL background on the Ang2 infusion model. Note the incidence of aneurysm formation in their hands, like what we've seen and Alan has seen, 90%, dramatically reduced by deleting this particular gene. Here are their diameter data and the weight data, once again very similar to what I've shown you with deletion of P47-FOX. This is the aneurysm classification scheme that Alan has developed, and basically what you're seeing is a shift to the left, meaning that in the absence of the PGES1 gene, there were either no aneurysms or much less complex aneurysms seen. And then finally note the impact on survival. They saw significant mortality in their experiments, more than most of us see, but nevertheless, you can see the mortality levels significantly blunted by deleting this gene. Now, what was most interesting to me was, in the wild-type mice, first of all, when they infused angiotensin II, they saw a large increase in the expression of the microsomal PGES1 synthase gene, and this was fairly specific in their hands. Note, of course, that when they looked at the knockout mouse, they couldn't see any expression as expected. What was most intriguing, however, was that in the presence of the Ang2 infusion, there were large increases in oxidative stress, as noted by the isoprostane formation, and this was completely eliminated by knocking out the PGES1 gene. And so this suggests that this particular gene is involved in oxidative stress in response to Ang2 infusion, and this was further suggested by their nitrotyrosine staining, which was markedly increased in the presence of Ang2 infusion in their LDL knockout mice and severely blunted by deletion of the microsomal PGS gene. And so we don't really understand the mechanism, but it may result from a shift in the icosanoid metabolism more towards PGI2 and perhaps PGD2 that have profound impacts on antioxidant gene expression and other anti-inflammatory pathways in the blood vessel wall. Now, Gib Upchurch's group has recently published a paper suggesting that manganese SOD may be involved in aneurysm formation. Manganese SOD dismutates superoxide to peroxide in the mitochondria. The peroxide, in turn, may have many effects in the blood vessel wall. And so they have used the elastase infusion model and shown a selective induction of the MNSOD within 1 day after infusing elastase as compared with the saline control. And you can see the induction was observed both at the mRNA level and at the protein level. And again, this was fairly specific for manganese SOD. They did not see this with the other isoforms of SOD that they looked at. Likewise, there was an increase in MMP9 expression in the same time course, essentially, as they saw with the manganese SOD. And they did some experiments in vitro and showed that if they gave Tempol to aortas, and Tempol's an SOD mimic, this could actually induce MMP9 and MMP2 expression. And catalase, which decomposes peroxide, could block this. So they speculate that the manganese SOD may actually be proaneurysmal by triggering an increase in MMP9, although this remains to be shown experimentally. So for the last few minutes, I'm going to talk now about our work that's been going on in myeloperoxidase. Myeloperoxidase is a heme protein that's present in very high concentrations, particularly in neutrophils and in humans and in human monocytes, to a lesser extent in mouse monocytes. Now, myeloperoxidase is very much involved in conversion of intimal macrophages into foam cells, and if you look at human blood vessels in aorta, it's clear that myeloperoxidase is a key enzyme involved in oxidizing LDL and causing it to be taken up into foam cells. What was most intriguing to me is the studies that have come out in the last decade, in particular this study that was published in the New England Journal, linking myeloperoxidase levels in humans that report to the emergency room with chest pain as being predictive of future cardiovascular events. Furthermore, there's evidence that myeloperoxidase levels are increased in patients with coronary disease in proportion to the severity and the acuity of their disease. Patients with coronary disease who have unstable angina have higher myeloperoxidase levels than those who come in with stable angina. Likewise, MPO levels can predict cardiovascular events in patients with peripheral vascular disease. So this enzyme may be involved in converting an otherwise stable lesion to an unstable lesion, for example, and this may be pertinent to aneurysm disruption or dissection. So myeloperoxidase is an interesting enzyme. It basically uses hydrogen peroxide and chloride anions, and it forms this very powerful species, hypochlorous acid. Hypochlorous acid chlorinates tyrosine residues in many proteins, and this is nice because it gives us a very useful marker to look for evidence of hypochlorous acid-mediated oxidative stress. Also, there's a convenient way to investigate the role of this process in pathological conditions by using a very simple tool, taurine, which is one of the most abundant amino acids in our bodies. It's sort of unclear what taurine does. It's not used as a building block for other proteins, but it's present in abundance. It's non-toxic. It can be used in humans. And importantly, it reacts with and detoxifies the hypochlorous acid, forming taurine chloramine. Not only is taurine chloramine not reactive like HOCl is, but it actually has powerful anti-inflammatory properties. It inhibits leukocyte migration and inflammation. As I mentioned, taurine can be given in high concentrations to humans safely without side effects, and it's been shown in a number of studies, for example, mostly in endurance athletes, to actually protect against oxidative stress and DNA damage during heavy physical activities. And so we decided to see the effects of taurine using the elastase model that Rob Thompson has been so nice to collaborate with and assist us. And I'm sure he'll be glad to see this data, which is finally about done. In any event, we took our mice, the 3 groups, control that were given heat-inactivated elastase and no taurine, the experimental group given elastase infusion and no taurine, and finally the treatment group in which we gave taurine for 1 week at 4% in the drinking water and then continued it for 1 week post-elastase infusion. And I'll mention the dose we chose has been studied in other people's hands in murine models, is safe and effective, and this is where we started. First of all, we confirmed Rob's data showing that in the elastase model, about 3 days after you infuse elastase, you see a large increase in myeloperoxidase activity in the aorta, consistent with the influx of neutrophils into the aortic wall, which he has previously shown. And so that confirmed then that myeloperoxidase was upregulated and would be present during this time. Interestingly, when we gave our animals taurine in the drinking water, we were able to totally suppress the formation of aneurysms in the elastase model, as shown here looking at the average diameter of the aorta at 14 days after infusion of elastase. To look at the mechanism, we've performed immunostaining for chlorotyrosine. As I mentioned, this is a very nice marker for myeloperoxidase-mediated oxidative stress. In the upper panel, this is the heat-inactivated elastase control aortas. In the middle panel, this is the elastase-treated aortas. And in the lower panel, these are the animals that were given taurine and infused with elastase. On this side, you're seeing the control immunostaining, and on this side, the chlorotyrosine immunostaining. I hope you can see, it's a little faint, but there's brown staining largely in the adventitia here, but also throughout the other layers of the blood vessel wall in the animals that were treated with elastase. And when we co-treated with taurine, we were able to totally block the formation of chlorotyrosine residues in the aorta, thereby confirming that we suppressed myeloperoxidase-mediated oxidative stress. We also looked at macrophage infiltration. I apologize, the quality of these slides is not very good. I was making them up just before I left. But clearly, we could see evidence of significant macrophage infiltration into the aortic wall in the elastase-treated mice, and this was also essentially abolished by co-treating with taurine. And this suggests that, as Rob said earlier, the influx of PMNs probably triggers macrophage infiltration, and this may in part be mediated through the myeloperoxidase. We also have looked at MMP activation, and this is the heat-inactivated, the elastase, and then the 4% taurine plus elastase. You can clearly see evidence of activation of MMPs here, 2 and 9, and this was fully blocked by co-treatment with taurine. As expected, when this occurs, we see a reduction in elastin degradation. Heat-inactivated here, elastase here, and elastase plus taurine. And I think you can see the inflammatory infiltrate associated with degradation of the elastin bands, which was largely abrogated by treating with taurine. We've also gone on to show that taurine works, although less effectively, in the Ang-2 infusion model. This is 1% taurine, and this is 4% taurine. And you can see this is not as effective. Looking at the incidence of aneurysms, it roughly is cut in half by treating with taurine. And this may reflect the fact that the role of myeloperoxidase may vary in these models and may be less impressive in the Ang-2 infusion model. Looking at the aortic diameter, you can see the data here, a graded response between 1 and 4% taurine and aortic weight, indicating that taurine is to at least some extent effective in both of these animal models. We looked at the complexity of aneurysms using the aneurysm scale that Alan developed, and in particular in 4% taurine, there was a large shift to the left, suggesting a less complex aneurysm in this model when we infused taurine. And then finally, we looked at oxidative stress by staining for nitrotyrosine, and these are the controls. Animals that were given the Ang-2 infusion developed lots of immunostaining for nitrotyrosine, and this was blunted by 1% taurine and even more impressively by 4% taurine. So to summarize, there's no question at this point that oxidative stress is locally increased in aneurysms. It's probably modulated by a number of pathways, including NADPH oxidase, PGE2 synthase, myeloperoxidase, and perhaps INOS. We can say that conventional antioxidants like vitamin E are probably not very effective and shouldn't really be investigated much further, but I think our evidence suggests that focusing on things like taurine, which may be targeting a more important enzymatic pathway here, seems to be effective and potentially could represent a safe, inexpensive, and efficacious therapy for humans with this disease process. Of course, targeted pharmacotherapy against these particular enzymes should also be investigated as a strategy for treating aneurysms. So I would finally like to thank the people who have been collaborating and participating, in particular my lab now at Cincinnati, Andra Blomkons and Bonnie Neltner. Back at Iowa, a number of people who I'm still collaborating with, including Francis Miller and Mike McCormick. I mentioned Alan Dougherty and Lisa Cassis at Lexington, Rob and Terry Ennis, who are collaborators in St. Louis, and then Minesh Thomas was a fellow at Iowa who is now at the University of Tennessee. So thanks for your attention.
Thanks, Neal, for a fantastic comprehensive overview. We have time for a couple questions. Let's start over here, Carlos.
I'm sorry, I'm running a little bit late.
No, absolutely. We're good.
A question from an epidemiologist. Which foods are rich in taurine?
Well, I mean, that's a question that I guess would take some time to discuss. I think the point of this is probably nothing is going to be rich enough to give the levels that we were giving in our animal studies here. I think for anything to probably be very effective at blocking myeloperoxidase, it's going to require pharmacological levels of taurine. I don't think you're going to get this from the diet, most likely, is the answer. And so we're interested to try to figure, can we give enough taurine here, and will it be well tolerated and effective? So anyway, that's my answer. I think it's going to take pharmacological doses. But the interesting thing is you can see that the taurine reacts with the HOCl right at the site that the inflammation is occurring, and it's generating anti-inflammatory molecules right there. That's why I think this is potentially an attractive strategy.
You can tell a lot of the sports drinks people have glommed onto the idea in terms of the endurance athletes, because you can find a lot in these formulations, these sports formulations.
Neal, do you think the neutrophils play is an important part in human aneurysm pathogenesis as they appear to play in the experimental aneurysm pathogenesis?
It's a really good question. It's probably going to be hard to answer since we don't get enough early specimens in the humans. I guess what I would say, number one, is not only can the MPO come from neutrophils and from macrophages that are there, but it can also be taken up into the blood vessels from the circulation. I don't know if you're aware, but myeloperoxidase does circulate as a protein and can be taken up into the blood vessels. So it's kind of complicated. It's not enough just to look for the cells, I think, is what I'm trying to tell you. Number two, as you're aware, these sudden triggered events may be at times where there's very intense PMN activation at a very brief moment, and then the PMNs are short-lived. And so it may trigger a destabilization. This is why I tried to hint that we think in acute coronary syndromes this is important. And it may be important in deterioration of aneurysms or perhaps rupture or dissection in the same way, that you may get a very brief episode of intense inflammation or recruitment of PMNs that may trigger a destabilization. And that would be hard to detect. There must be some molecular imaging tools. Maybe we'll hear more about that during the subsequent talks.
So I have a question for the experimentalists out there. In terms of the differences potentially between the two models, both in terms of pathophysiology as well as responsive therapy, can you tell us in your hands, first of all in the elastase model as well as the angiotensin II infusion model, what the background of the mice that you generally use, what's the diet, and any other manipulations?
All of these that we have been studying are in the C57 background. Obviously, in the Ang2 model, we have put them into a hyperlipidemic background, and we use both the ApoE and the LDL models, and we seem to get pretty much equivalent results. All of these studies were on chow diet, so none of these were in high-fat diet or any other manipulations except just a standard chow diet. So I don't know. I think there's many different ways to do it, and that's the exciting part about the whole thing, to try to look at interactions between these variables as well as the mechanical factors that we've heard about.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.